I work on many different things, and I worked on different things, and over my career. You're right, I didn't mention about the energetics in my web page or my NIH CV. It's not there, but I wanted to start my talk with that. And you know, for a long time, actually, when Jay and before Jay and when Jay joined, you know, my group, you know, I was very much interested in this particular phenomena here of energetics and how it actually functions in you know, driving organ function. So here's a very generic term, work, and of course in muscle this is contraction, in brain this is all the processes that the brain does, predominantly related to establishing the, you know, ion gradients that are responsible for electrical activity in the brain, etc. But this you know, this essentially, this diagram, you know, really applies with respect to this work and coupling work to, you know, production of ATP. And you know, in a sense, I must confess, you know, why was I interested in that? I was interested in it because I thought I could do something about it, not because I thought this is a really interesting phenomena, which it is an interesting phenomena, but I really thought that we had some unique tools in the, you know, early or late 70s that we can actually tackle this problem in vivo in intact systems, which started my career to working with E. coli. I had to learn, you know, I'm a physicist by training, and then I had to learn how to grow E. coli, and I made significant contributions about how to harvest E. coli, actually, which remain unpublished. I guess I should do it one of these days. And but at the time that Jay came to work with me, and we had moved from E. coli to working with the heart. And again, I have to confess, I mean, that I was being an opportunist in working with the heart, and not because I'm a cardiologist or, you know, I was really you know, interested in the fact that, you know, you can actually take a heart, take it out of the animal, put it on a perfusion system, and it works. And you can actually control a lot of the physiological parameters. You can change its workload, you can change its, you know, the pressure against which it works, you can change its heart rate. And so it was an ex vivo system you can put into a relatively small, in those days relatively big, MR tubes and study. But very soon I moved beyond that, and we were working in a really superb collaboration with Bob Bucky at the University of Minnesota, who superb scientist who just recently retired, working with instrumented animal models. And we were doing experiments like this. These are actually, this, I don't remember, I think this is a dog, but it could have been a pig or dog model, where we were looking at, we had developed some of these techniques for acquiring phosphorus spectra with spatial resolution across the heart. So you can actually see these are labeled epi, mid, endo. These are layers across the left ventricle. And this is a phosphorus spectrum. This is creatine phosphate. These are three peaks are coming from ATP. There's a peak here that shows up during this perturbation, which is you know, kind of it's a flow restriction, and then the heart is made to work harder under flow restriction. And this is an inorganic phosphate. So these are all of those molecules that I showed in the previous bioenergetics diagram that you can detect it in a phosphorus spectrum. And so you can see here that we know that the heart's response to workload and blood flow, etc., is non-uniform across the heart wall. And these were really very exciting experiments. We can actually see that response in the energetics differentially across the heart wall, going from the superficial layers, which don't do as much work, going to inner layers where the workload can be much harder and causing, under flow restriction, causing much more severe ischemic events. Then we went to intact animals. This is again a paper where Jay is a co-author, and this was done by one of our students who worked really with Jay, and showing that we can do the same thing in the intact animals. So that was a open chest preparation, and this is now a closed chest preparation. It's not totally uninvasive because we actually, just to show that we can do this, we surgically inserted a bag filled with phosphoenol pyruvate here, which doesn't exist in the heart or the skeletal muscle. So you can actually see that we can differentiate peaks where you can see here's the phosphoenol pyruvate in this voxel, but here it's not there, it's not there, it's not there, it's not there. So we can localize it very nicely and do intact animals. So we were thinking, okay, that's great. Let's go to a human. And but what is unique about these things is that these are animal experiments. They are done at very high magnetic field. So this was done at 9.4 Tesla. The previous one is 4.7 Tesla. This was at the time that people were using 1.5 Tesla was the standard clinical instrument. So we started looking for a high field system. And about the same time where UAB was looking at a high field system, actually, and so I ended up getting a 4 Tesla magnet from Siemens. This is this magnet. It looks like a very low tech structure. It was such a long time ago, actually. I don't think color photography was discovered at that time. But this magnet, so we actually were driven towards developing that program really because of spectroscopy and the work that I was doing in bioenergetics, which Jay now says you don't have it on your CV anymore, on your webpage. In fact, recently a colleague from Oregon came and he was asking me about, Camille, I want to talk to you about this bioenergetics, about this molecule, that molecule. I told him, Charlie, I don't work in that field anymore. And so, and the reason is, when this magnet was essentially ordered, we're waiting for it. There was this development where Seiji Ogawa, my colleague from Bell Labs, showed that if you change the animal's oxygenation state, you can actually see changes in the images. And that led to the discovery of functional imaging that Jay mentioned. So we were really waiting for that magnet to come to Minneapolis to do this functional imaging experiments, which we did and published in 1992. It came at the same time with a paper from MGH. This is the work from MGH. The two papers were submitted within 5 days of each other, by the way. And for us, unfortunate thing is that we submitted our paper to Nature. I didn't want to. My colleagues really wanted to. And Nature rejected the paper. So this is now extremely highly cited paper. I don't know, every year it gets hundreds of citations. But so don't feel bad if Nature or Science rejects your paper. I published in Science and Nature, but compared to this paper, they are nothing, the other papers, but Nature rejected this paper. But what is interesting is that this is our work, and that's MGH work. There's a big difference in the quality of the functional images. This is a functional image obtained in the human brain showing visual activation. And we know this is approximately the visual areas. Here, there's some blob of activity here, but here it kind of follows some contour. There's some structure here. It follows the gray matter, actually, in the functional image. So if you look at it this way, here's the anatomical image, and here's the functional image. You can kind of put an outline of the gray matter areas. Here's the sulci, and this is the CSF space. You make an outline and you put it on the functional image. You can see that, well, okay, it looks like there's some very large activity on the surface here, not in the gray matter, but quite a lot of activity in what is now the gray matter. And this is essentially, it took us a long time to understand it. But the difference between the blob images and this image, why we see this, we actually expect, we don't expect this. We were unhappy to see that, by the way, but we expected to see this, and it turns out that actually happens because we were working at 4 Tesla. So the magnetic fields, which we were interested in for spectroscopy reasons because of SNR, were also, turns out to play a very critical role in functional imaging. Essentially, we published a paper that said something like the yellow, utilizing high-resolution images, we see 2 distinct regions ascribed to be microvascular and visible venous vessels identified as giving rise to the fMRI signal increase. So essentially, we are in fMRI, when we do brain functional imaging, we are looking at secondary signals, or tertiary, if you like. There's a neurovascular coupling, there's neuronal activity, there's neurovascular coupling that leads to increased blood flow and metabolic changes, and that causes changes in the deoxyhemoglobin content and blood volume, and that then causes the functional imaging signals. And of course, vasculature then, from just what I told you, tells you that the vasculature is very critically important. So if you look at the vasculature, this is brain surface, the peel surface has these big vessels, veins and arteries. Then you have these cortex here, this is white matter. This is the, approximately 2-3 millimeter thick cortex, depending on where you are on the brain, thickness varies. But, you have these blood vessels, diving arterioles, and then veins that return the blood, and then the capillaries. So all of those are actually important. And it turns out that the signals that I was showing you, I mentioned to you that the highest signal was actually coming outside of the brain. It's because of the fact that, we were seeing some of this associated with, the blood volume and blood deoxyhemoglobin associated with the capillaries. But a lot of it we were seeing because the deoxyhemoglobin changes are not static. They flow and collect into these veins, and then they collect into these peel veins, so that we are seeing it outside on the cortex at the peel veins. But our 1.5 Tesla colleagues, it turns out, they were only looking at this. But at least at 4 Tesla, we had this plus this. But this is not a very good situation for functional imaging because of the fact that this is not a very accurate signal. There aren't that many vessels. Their distribution in space is not very high. And so if your spatial resolution is, I don't know, say 5 by 5 by 5 millimeters, this is okay. But you can do that with PET. And we were interested, can we do high resolution imaging? So there we are interested, we want to be able to tell this apart from this. But not only that, we really have to be, for example, to understand brain function, tell this apart from that. Because this cortical thickness is not a uniform entity. You have situation, for example, if I put this is the peel surface, this is the white matter, there are at least so-called 6 layers with different neuronal types. And not only that, if I get this putative diagram here, you have connections to different layers of the brain coming from different parts. For example, thalamic input comes to the middle layers, but then you have cortical connections to the superficial layers and the inner layers, and there's a layer-dependent circuitry. So if you are just looking at the peel vessels, that kind of resolution, okay, it's still a lot you can do in the human brain, since we don't know too much about it, but in some ways very inadequate for understanding brain function. And Jay mentioned the Brain Initiative, and when the Brain Initiative was announced, there was an article by Tom Inzel, Story Landis, and Francis Collins. He was the head of NIMH, she was the head of NINDS, and Francis Collins is still the head of NIH. But what they said in this article, announcing why we were, we need the Brain Initiative, one of the things they said, well, they said human neuroimaging captures the whole brain in action, but each 1 millimeter voxel includes at least 80,000 neurons and 4.5 million synapses. So, in a sense, it's a big advance to be able to do imaging in the human brain, but it's a lot of neurons doing multiple different, capable of doing multiple different calculations that we are averaging. So we have to get to high resolution, high specificity, and so from it was not the Brain Initiative actually. I think Brain Initiative finally caught up with me, but when the functional imaging came into existence, we were in fact very much pushing this area, trying to see how we can go beyond this and look at those things. And the answer, it turned out, of course, a lot of pulse sequence development, but also high magnetic fields. And where does that come from? So this is from our 1993, it was a second paper on functional imaging, showing our modeling of functional imaging signals, what we think is happening in terms of the signals we are detecting. So it's actually interesting that we are looking at blood, radius. These are essentially infinite cylinders. In our mind was sort of a first approximation to blood vessels of different radius. So capillaries are somewhere over here, and and then you go up to much bigger vessels, and this would be the change in so-called T2 star or 1 over T2 star. This is what we are detecting in functional imaging in terms of an MR parameter that would come from essentially changes in the deoxyhemoglobin content of these infinite capillaries. And you can look at this diagram here, these numbers, as the deoxyhemoglobin is increasing. And but you can also look at it as deoxyhemoglobin staying the same, but the magnetic field is increasing. It's the same thing. It's actually, the effect comes into it as a product of deoxyhemoglobin concentration and magnetic field. So one or the other doesn't make doesn't make any difference. It's the product that is important. But what was also very important was that there are two regimes here. One regime is where effect becomes independent of the radius. Another regime where the effect gets smaller, but it becomes actually in the area of capillaries. And that's essentially because diffusion of water averages are around the effect of the magnetic field inhomogeneity is caused by deoxyhemoglobin. And that averaging reduces the effect, but it also allows you, it turns out, a mechanism by which you can kind of inhibit this and promote this signal in the functional images. And then we looked at how this different regions come into the effect. And this is small. It's right, actually. Let's see. So, okay, so that's the capillaries, and these are the other ones are the big vessels. But here's the magnetic field expressed in frequency units. So the big vessels come into the problem as a linear effect of magnetic field. They go up linearly with magnetic field, but the small vessels go up as quadratically with the magnetic field. So we were working at 4 Tesla, so we actually, these modeling paper, kind of gave us an understanding of why 4 Tesla images were so much better. And then we decided to go to then higher magnetic fields, and we established the first 7 Tesla in 1999. This is my colleague Mike Garwood sitting in the bore of the magnet, and at that time. And Jay mentioned the importance of engineering, because we really had to put this instrument together ourselves. We were in a very good relationship with this magnet company, was led by a very visionary leader at that time, he really bought into our idea, and we didn't have the money to pay for it at that time, but he said, okay, he'll build the magnet and give it to us, and we can pay it when we can, if we can. And we paid him. I actually submitted a grant to NIH. It's a very unusual grant, because the magnet was there. I submitted an NIH grant. I said, this magnet is there. We should not punish him for taking the risk. He facilitated. If you think that this is a good science, please fund this grant so I can pay him. And they funded the grant. And So I paid him. So then we did several magnets like that. Actually, we did a 9.4 Tesla, etc. And then he was bought by an American company. He made millions, but the American company said no more, none of those deals anymore. You put the cash, and then we give you the magnets. So that's where we are now. That was Agilent, but anyway. So 7 Tesla, and we showed that 7 Tesla actually produces much better functional images than 4 Tesla, as we expected. These are as a function of so-called echo times. Long echo times, 7 Tesla doesn't do very well. We understand this. There's an optimal echo time depending on the magnetic field. It's like around here for 7 Tesla. It's around here for 4 Tesla. And with that, we were able to show for the first time that you can do these orientation domains in the human. These are structures, organizations in the human visual cortex, in the visual cortex of also many animals. Not rats, by the way, not rodents. Rodents they have these cells that are orientation sensitive, but they are not organized in maps. But animals like cats, etc., monkeys, and they have these organized cells which respond to different orientations. So each eye divides into different regions in the brain. So one eye versus another eye. These are called ocular dominance columns. And then superimposed on that are the orientation domains. And we were able to show, after a lot of really both engineering and methodological development, we did a lot of instrumentation, a lot of methods, understanding of the signals, why, where are they coming from. We were able to show, this is a 2008 paper, 2007 and 8. In one individual, you can map the ocular dominance columns. These are right versus the left eye. They look like, I showed you this columnar organization in a different orientation, but on the surface of the cortex, they look like these fingerprints, and they have the right periodicity, etc. And on the same individual, we were able to map the orientation domains, these color-coded patches, which each color represent essentially, groups of ensemble of neurons that are activated when they are encountering or looking at a particular or gridding with an orientation. So that at that time, those kinds of images of orientation domains you can actually obtain with optical imaging in the monkey, because monkey cortex is visual cortex is easily accessible on the surface, so you can actually do optical imaging with a window, putting a window in the skull, and they were able to obtain these dark lines are the ocular dominance columns, the colored lines are the orientation domains, so these are the so-called pinwheels. A lot of information exists about what is the distance between these pinwheels, so-called these, I forgot now what these regions are called. I'm skipping the word, but they're perpendicular to the orientation domains, etc. And this is what it looks like in the human brain. It's not as high a resolution, but we don't need a window in the skull, and we can do it whether the brain is perfect or folded. We don't need a smooth brain to access that. And the numbers actually, in fact, are in excellent agreement with the monkey data. But again, let me come back to the technology perspective again a little bit. So we did a lot more modeling afterwards. So this is different than the modeling I showed you initially, which was modeling what the relaxation effect would be if you take tubes and then you get signal changes in the relaxation parameters of water. But this is actually functional imaging modeling. You actually take into account, well, what is the blood volume of capillaries? What is the blood volume change? What is the blood flow, oxygenation levels, hematocrit, and everything? And you put that into a model, and you come up with fMRI prediction about, these are so-called spin-echo fMRI, where you are much more sensitive to these vessels as opposed to these vessels. And you can see again the vessel, the magnetic field dependence. So with spin-echo fMRI, these are weak signals. Most people don't use them because of the fact that they are weak signals. You use them if you are interested in columnar level organization. But they are increasing rapidly, the small vessels with magnetic field. They tend to level off beyond about 10 Tesla or so. There are two limits here because there is some controversy about where the blood volume changes in the brain when there's neuronal activation. Is it in capillaries? So this assumes no capillary blood volume change. We are probably closer to this than to this one based on some optical imaging data, but this is a controversy. And here's what happens to these PL vessels. They actually kind of decrease with magnetic field after some increases. And you can guess now we are working at something like 10.5 Tesla because of this graph. But this does not take it. This is just the functional imaging effect. Doesn't take into account the fact that signal to noise also increases. And if you take that into account, so-called you calculate functional contrast to noise ratio. This is how it depends. And these are again these two different assumptions. But remember. That this is actually normalized to 1.5 Tesla. At the very beginning, there was 1.5 Tesla and 4 Tesla. But compared to where we are at 7 Tesla, we are almost 30, 40 times better, if not more. And then at 10.5 Tesla, we expect about 100 times better signals. So this is the reason we have been pushing high fields. And so we have supralinear gains with magnetic field in this bold contrast. But in addition, we have higher signal-to-noise ratio. And then we have higher spatial accuracy because we can actually restrict the signals to these capillary as opposed to the rest. And I can tell you, I don't want to go too much about this, but the first at 7 Tesla, the first cognitive, or if you like, human brain experiment we did was not the ocular orientation domains, but it actually is tonotopic mapping. Tonotopic mapping at that time, 1.5 Tesla people they couldn't produce a convincing tonotopic map. Whereas in the animal model, we knew that there were mirror symmetric tonotopic maps, and likelihood that the human brain would have something like that was very high. And the reason we surmised that there was a problem with 1.5 Tesla functional images, so we did the 7 Tesla, and sure enough, we showed these mirror symmetric tonotopic maps that were highly reproducible, published in 2003. But I can tell you in a summary of kind of a decade of work, actually, since then, of really instrumentation, methods and everything. What we have done on the auditory system. So 2003, we showed that there are mirror symmetric maps, tonotopic maps. And then, there was quite a bit of hiatus in tonotopic mapping. Then we came back 2013. We actually published the fact that the mirror symmetric tonotopic maps we can also image in the inferior colliculus, because there are all these regions in the brain going from the ear to the primary auditory cortex, which are involved in auditory processing. So this is a much smaller structure. You can see that compared to that. It's in the brain. It's a very small structure, but because we have this high specificity and high resolution, at 7 Tesla we can map that. And we actually showed in the same paper not only that there's a tonotopic mapping, but superimposed on it, there's a bandwidth mapping. Actually, the neurons have, as a function of space, the bandwidth that they respond changes. And then 2015, we went to medial geniculate body. It showed that, there are actually these two tonotopic maps in the medial geniculate body, and we are now talking about columnar level mapping here. And then, we went back to the auditory cortex. This is, we have actually more recent papers than that. The slide I prepared some time ago, I keep using it. But, in 2015, we published a PNAS paper showing that when you look at the auditory cortex, we can actually, we can do it with high enough resolution so that we can separate different cortical layers. So we can actually, the cortex is, as I mentioned, 2 or 3 millimeter sheet. It's curved. We flatten it, and we can see the responses across the layers, and there are regions. It turns out in the primary auditory cortex, mainly across the layers, the responses are uniform. In other places, they are not. But it was also very interesting. In the same paper, we showed, coming back to the layer specificity, we had postulated, well, okay, we can actually see them. They tend to be uniform, but our postulate was, if we can modulate them by attention, most likely the modulation will be layer specific, because modulation with attention has to come from cortical-cortical connections, and that we would expect them to come to at least outer layers and maybe in the inner layers, but certainly not in the middle. And in the same paper, we in fact showed that we were able to ask people to either pay attention to the auditory stimulus or we showed them a visual stimulus, and then they were asked to pay attention to the visual stimulus. And the biggest responses we saw were in the outer layers, essentially. Let me just show you. I have to kind of turn that around. So this is the white matter layers over here, peel surface over here, and the outer layers over here, showing that you get more in the outer layers. So this is one path we have taken in the brain, and but there's another path. I'll briefly mention it, and some of you may know that we've been working on the so-called Human Connectome project, where we are not so much interested in very high resolution. I mean, we are, but we are not there yet to do whole brain connectivity. And that Human Connectome project aims to essentially map connections in the human brain in the as high a resolution as we can get. So in the millimeter, it uses 2 MR technologies. One is so-called resting state fMRI. I'll tell you what it is, and diffusion-weighted MRI to infer structural connectivity. So the latest paper we published from that is this parcellation paper, and that came from our consortium, and Nature called it Brain Redefined. It's actually far from being redefined, but it was good step in some people are interested in this parcellation. I'm actually interested in parcellation at the level of a few thousand neurons, which is way beyond that, but we will get there, I'm sure, in my career, I hope. But just to give you an introduction of these technologies, so when you do functional imaging, you acquire a series of signals, and then you ask the person to do a task or you present a stimulus. But if you don't do that, you just allow the subject to rest in, that's why it's called a resting state, rest in the magnet. There are no input, you don't ask them to do the task, they're just lying in the magnet, and you see these signals are fluctuating. These are 3 different regions in 3 different color, blue, this light blue, green, and red, and these are, 4 regions maybe. And if you look at them, they are all oscillating at about the same periodicity, and they are correlated. And if you map those correlations, so you can say, okay, give me the image of everything that is correlated with something over here or in the middle, you get these things in the brain which look like activation, but they are not. They are just correlated temporal dynamics. And it turns out that these areas respond on brain networks. So areas of the brain that work together, that form networks, oscillate together. It's interesting that these oscillations are not only seen in fMRI, but if you do 2-photon optical imaging of columns, you actually see them too, oscillating just like the fMRI signals. So these networks, so somehow it's a fundamental principle of brain organization. So this is like this area, you choose a point, and you say, give me all the correlations, positive and negative. So these areas are correlated positively, blues are correlated negatively, and you can move the point and get new areas. So this is essentially so-called functional connectivity. There are many ways of looking at this, and the human connectome data produces a lot of results from this. And in fact, I didn't put it on my slide, but recent work we were doing with Fudan University, actually. We actually showed that there was following work at Yale. We showed that you can take these functional connectivity maps of people, and you can identify individuals with something like 99% accuracy based on these functional connectivity maps. But what I wanted to show you, since you are an engineering department, a little bit about the fact that, again, why engineering solutions are important. And we did these experiments both at 7 Tesla and 3 Tesla, the Human Connectome project. I mean, we generated a database of 1,200 people at 3 Tesla, but 184 of those people were scanned at 7 Tesla at the same time. Not at the same time, but they were scanned at 7, the same people. So this is what we would call contrast to noise ratio in being able to obtain such measures of these correlations or fluctuations. And not correlation. Correlation itself is like a functional connectivity, contrast to noise ratio. But, you know, this is on the scale of 3 Tesla, what it looks like, and that's what it looks like on the same scale at 7 Tesla. So in fact, there are really significant gains in the 7 Tesla data with respect to identifying those networks. And these are the subcortical areas, and this is the cortex somewhat flattened. And then you can ask, like we did before, you know, you can put a seed, and the seed is here, right here, in this case, in a subcortical area, subcortical nucleus, putamen in this case, and you can ask, okay, how is this connected to cortical surface and these other areas? Well, this is 3 Tesla data, and by the way, the 3 Tesla data of the Human Connectome project is really much more advanced than anything else out there, and this very advanced 3 Tesla data has a hard time telling this connection to us. It is kind of like noise. It's approximately somewhere over here. And then if you do the same thing on 7 Tesla, and you can actually see this connectivity very beautifully. So the contrast to noise ratio is much better. You can do this much better at 7 Tesla. And this is reproducibility from time to time, and much higher at 7 Tesla than at 3 Tesla. But 7 Tesla doesn't come for free, you know, and there are major problems at 7 Tesla. The physics of RF waves, you know, because we have a static magnetic field, and then we have an electromagnetic, oscillating electromagnetic field, the frequency of which changes with the magnetic field. So at 7 Tesla, we are working at 300 megahertz, and you know, at 4 Tesla, you know, something less, 180 or whatever. 3 Tesla, I think, is 120 or something like that. So when you are looking at these are human head maps of this RF field we are pumping in to excite and get those spins. The map, so-called transmit B1. This is the RF field. We always label it as B1. Static field is B0. And you can see that at 7 Tesla, they are extremely non-uniform. They vary from here to here, and then within slice, they are really non-uniform. And this is a problem. So for us to be able to do that Human Connectome project data over the whole brain. So if you want to just look at the visual cortex, okay, well, we can forget about these areas and we just focus around here, and well, they're more or less uniform. But if you want a map all the way from here and lower, the cerebellum is not shown here, it would be over here, to here, there's this huge inhomogeneity. And so really there had to be a lot of instrumentation and physics development. And the problem comes because of the fact that when you go up at this frequency, RF frequency that you are using, you actually start running into a phenomena because of the fact that the dimensions of the object you are trying to image, like the head, becomes smaller, larger than the wavelength. And the ultimate, if you talk about the human torso, it's even worse. The dimensions are much bigger in the human torso. So-called relative wavelength is like 3.3 here. It's about, 1.5 or 2 in the human head. This is a different regime in physics. This is like when you turn on, if I turn on a light over there, that light wavelength is very small compared to this distance between me and the light and my body. And that has to propagate. It has to actually be a wave and propagate. So, in fact, what is happening, because this wavelength is small, these RF waves when you're working at 7 Tesla have to propagate from the source you create into the body. And that creates the problem. So this is actually a simulation of that propagation. So what you are doing is that here's the RF coil. You are seeing a cross section, just a sagittal section. Here's the human outline of the human body. Of course, you would not be able to map this experimentally like that, but at least not in a standard MR machine, but you can calculate it. So here's the RF coil, and when it is turning red, that means you are pushing current into the RF coil. It's generating these magnetic fields, and it has to, as a wave, propagate into the human body and from the surfaces, and then propagates out of the magnet like this. And then because of they are propagating, they actually can interfere with each other constructively or destructively and cause these inhomogeneities. So you can do very weird experiments with this. For example, here's that same picture but static. Here's the RF coil. And this RF coil generates a uniform magnetic field when the subject is not in there. It's uniform approximately the level of the physical dimensions of the coil. And you put the subject in it, as you've just seen, depending on the time point where you capture that, you see these non-uniformities, the very destructive interferences here. And you even see actually that the highest points in the human head, it's outside of the RF coil. So you can do an experiment, for example, which is a really, for most MR people, this would be a weird experiment. You excite with this coil, you then detect over here, outside of the excitation, right? Outside of the excitation. Most people will say that's crazy in the MR world. Yeah, but it's not crazy because these are waves now propagating in the human body. And they go from, they enter on, if you like, they enter on your toe, they come out of your head and bounce back from the border of the, where the skull is, they bounce back because there's a magnetic electrical properties of the material is changing, air versus tissue. They are reflected from this boundary, and they form this high intensity. So you can get images. Like you put it at 10 centimeters, no problem, there's this distance. 20, 36, 60, you are like about this much out of the coil where you are pumping the energy, and you get images. Really weird physics phenomena. But, and then that was the reason actually initially when 4 Tesla came into existence, people actually noted this, not this particular phenomena, but that there were non-uniformities. This is not from our lab, but when actually this is from the Philips group in Hamburg. They showed, or maybe the Siemens group, but anyway, Philips group and the Siemens group and even GE group, they all showed these results. If you obtained images at 4 Tesla at the time, they look terrible compared to 1.5 Tesla images. They are the center bright, which is what I was showing you as a map, and torso image. How terrible this torso image. You cannot even recognize that it's a torso image. It's coming because of what I'm explaining to you. At that time, people did not understand this. And lucky for us, because the companies then decided, oh, this is too tough. It's not going to be so easy. Why bother invest in it when we are making money from 1.5 Tesla? So they gave up their 4 Tesla equipment. One came here, one went to University of Minnesota, one went to NIH. And ours, when it came, we did the fMRI experiment on it, as I mentioned. But, so we understood this problem. So we were saying, okay, how are we going to be able to solve that problem? Actually, you can solve it. There's both a methodological and an engineering solution to it. So the concept of multi-channel transmit. So instead of transmitting these coils, these waves as a uniform coil, we then actually break up the transmitter into multiple pieces. So these are 16 individual transmitters. So we had to duplicate our transmitter system. Normally an MR machine has a single transmitter. Ours has 16 and now 32 transmitters. And this is a recent demonstration looking at 2 slices, and then we developed methods for generating RF pulses that actually optimally use this. So, for example, these are 2 slices and mapping so-called flip angle that is essentially proportional to the RF B1 field. It's very non-uniform, just like what I showed you. Higher on the top of the head, lower at the bottom of the head, in-plane non-uniformity. But using this kind of a coil and these new RF, so-called parallel transmit RF pulses, in this case so-called one spoke, we can actually get them to be non-uniform. So as a result of all of this physics and engineering development, so at 4 Tesla, body imaging started like that. And then we solved that problem with the brain, and we said, why not go to body imaging? Why not go back to heart again? So this is what we can do at 7 Tesla now, when at 4 Tesla, you couldn't get a decent image. But it's only possible because of this parallel transmit. So many of you, if you are a cardiologist and you look at cardiac images, you can say, well, that doesn't look really so much better than what I can do at 1.5 Tesla, actually. And indeed, it doesn't, but this data has much better signal to noise. So you can ask yourself, what can I do with that signal to noise? You can do things like, one of the things you can do, something we showed is angiography without contrast agents. This is angiography without contrast agents. And this is actually some very early work on the body imaging. And if we were to go back and do this, we can actually do significantly better than this at 7 Tesla. For example, the transplant people are very interested in this, kidney transplant people, because of the fact that they don't want to give contrast agents because of their nephrotoxicity. Just to give you a comparison, so this is our early 7 Tesla, and this is the, if you like, gold standard X-ray based angiography. So we're getting there. We're getting there, and I bet you now we can do quite a bit better than that. I don't want to talk about that, but here's, for example, just to show you, we understood a lot more about. We have to go to other RF coil designs. RF coils that we normally use in the human body at 7 Tesla, not in the brain. Brain is okay, but in the body, the size is even bigger, as I had mentioned. So we have to go to antennas as opposed to RF coils, which are normally like loops. So this is a dipole antenna. So we combine a dipole antenna and a loop on a single circuit board. In this case, you can, modularly, you can go, this would be a 8-channel assembly here, each of these containing a loop and a dipole. And then you have 2 of those. You can put one of them on the subject, and you put them in a cover like that, and you put them on the subject, one underneath the subject, one above the subject. Of course, we have a lot of cables, that's, and then some of the most recent images now look like this for, kidney, prostate, cardiac. So one thing that, 1.5 Tesla does better, and we still don't do as well, but we don't care so much. For example, if you look at this image, okay, the target was prostate. So we actually optimized over the prostate for these coils to have the best region. And outside of the prostate, we get these destructive interferences. They're dark areas. But okay, we are not interested in these areas. So it's not, you can solve this by having more and more coils, but this sort of a target. So this is kidneys, you focus on the kidneys. Now the kidneys are looking nice. Again, heart looking very nice, and some more cardiac views at 7 Tesla. But as I was saying, okay, okay, we can get these nice images, but they have high signal to noise. What can we do with them? Well, you saw high resolution angiography, but we can go beyond that. This is, again, non-contrast agent perfusion measurement. So this is a very tough signal to noise dependent measurement. But because we have the signal to noise, so we can actually measure the perfusion, tissue perfusion, in this case kidney, with this kind of a resolution and accuracy without having to give any contrast agents at 7 Tesla. We can go beyond that. We can look at, again, this is enabled by this multi-channel transmit, looking at these so-called 4D flow. We can, in the blood vessels, we can see, exactly the direction. These are the flow streamlines. Color coding is the velocity of the blood. We can obtain these. You can do this at 3 Tesla, but we can do it at 7 Tesla at a much higher resolution. So, for example, it's not a big deal to map the aorta, as I've shown you. It's a big deal to map the aorta actually at 7 Tesla because it's such a challenging target because of this B1 problems that I showed you. But for example, you can look at, there's Pierre-François in our center has been working on aneurysms in the brain, where we are looking at very small blood vessels and aneurysms. So this is an example of a subject who has a small aneurysm, about 5 millimeters, and you may be able to pick it up in this case, right? Let's see, where is it? Right there, right, that guy over there, right there. And, well, I should have just clicked on this one. It just shows right over here. And it's about 5 millimeters. And we can measure, visualization is not a problem, but we can actually measure the flow patterns and the flow itself and calculate parameters like vorticity and these. These are this is looking at the same aneurysm with all of that different views. But we can, for example, we can do the 3-dimensional vortical structure within the aneurysm. And so it turns out that actually the aneurysm has much lower velocities, but much higher vorticity and shear. So, and the idea is actually people who are working on this, interestingly, both neurosurgeons and the aerodynamic people from the aeronautics department, where they are interested in flow, are working on this together with us and trying to understand, can they predict, for example, if they have find an aneurysm, whether that aneurysm is going to evolve to become a problem, should they interfere, or should they just let it go? So I told you a lot about kind of physiology and physics and some engineering, but as I mentioned, we are going to higher magnetic fields. This is our 10.5 Tesla magnet, which we just, actually it's a long project. It started in 2008, and we only recently finally got, 2 years ago we got to a field. About one year ago we got to operational status, and just recently we got the FDA approval as a investigational device. It's a very big magnet, as you can see. Humans are kind of standing next to it as dwarf, but very threatening in many ways. Actually, my colleague Mike Garwood, whose picture you saw in the 7 Tesla, when he saw this magnet, it was a weekend. It first came in on the weekend. I was there He came, Camille, did you see this? He said, oh no, I think we went too far. That looks very scary. But you put some nice touch up on it and color and things like that. It doesn't look so threatening. And we are actually now under FDA approval, we are doing 20 individuals where we are looking at them very carefully to make sure that we don't encounter safety. But we know it can work. These are pig images in the pig brain obtained at 7 Tesla. With interestingly, in electromagnetics, what is really important is not the dimensions or the wavelength, but the ratio. So the ratio is what is really important. So what we did for the 7 Tesla human torso is essentially now applicable to more or less 10.5 Tesla human brain in terms of how we are going to image that. So those coils that we developed with dipole antennas, etc., are now they are not good for the human head at 7 Tesla. They are good for the torso, but they are now good for the head at 10.5 Tesla. But we are very ambitious. We know we can do even torso imaging at 10.5 Tesla. So this is a 75 kilogram pig, and actually we have really nice images of the human torso as well. We are not showing them yet, but we do have them. And this is a volumetric image. You can see that there are some problems, but some of the most sophisticated techniques have not been employed yet, and so we think that we'll be able to accomplish this no problem. So let me. I just wanted to get to this slide at the very end. I had a few more extra slides there, but essentially I showed you a lot of things, but it really takes a big team to be able to do this. We have PhD electrical engineers, and we have mathematicians and working on image reconstruction, physicists working on pulse sequences, and we are all by somehow osmosis somehow interested in physiology, neuroscience, etc. But one of the most, a lot of people with many different talents contribute. And of course, for a while, when we were working on the heart, Jay was part of this big group doing very exciting experiments in the heart. But I want to especially acknowledge our, engineering group, who's composed of some real engineers, some physicists, who are kind of pseudo engineers as well, and without their input, nothing would be possible, because a lot of these instruments are first put together with us. Even when we work with Siemens, so I tell Siemens offers a commercial multi-channel transmit system now, 8-channel. You can buy it. We had a 16-channel by ourselves for a long time, and now we have 32 channels by ourselves. And so it's really important. I mentioned to you about the Human Connectome project. That was a major consortium effort between WashU and Oxford University and us, University of Minnesota. And in particular, I'd like to acknowledge David Van Essen, who was my co-PI on that. And Human Connectome project actually produced a lot of new imaging sequences, some of which you are using here. And they come as a result of the work conducted by these individuals. And that 4 Tesla magnet I showed you before Siemens discovered color photography is now a statue in our courtyard. Thank you very much. I'll end here.
So these are all lineages of Barbara Oppenheimer now.
Mm-hmm.
So we have time for a few short questions. Anyone? So I'll start the question about you are pushing the envelope of the first 3 Tesla, 4 Tesla, 7 Tesla, now 10 Tesla. You actually can measure the chemistry of the brain chemistry, that like compare normal or psychiatric patients brain chemistry with ATP flux. That's actually unique application of the super high field.
Yes. So that area I didn't cover very much. That's area. I mean, I started, in fact, with spectroscopy, but you can do a lot of brain spectroscopy. We do a lot of brain spectroscopy, and we can do some of the similar type of things we did on the heart. You know, together when you were there, we can actually, for example, measure ATP turnover in the heart and in the brain. We can do it. Actually, it turns out to be, for a lot of reasons, it's actually more straightforward measurement in the brain than in the heart. And you know, we can measure a lot of neurotransmitters like glutamate and GABA, and but spatial resolution is not as good as imaging. And in fact, I think that in a lot of brain applications, you know, the two can't come together. I mean, there are people who are interested, for example, in functional images or network you know, what's happening to networks activity, and you know, they show that, you know, things correlate with GABA levels and things like that. So it's an area that you know, we also are active in, and we expect the spectroscopy to get much better at 10.5 Tesla as well. And one of the reasons we actually came back to 7 Tesla cardiac imaging after I kind of left cardiac imaging was that once we solved the problem of doing how to do imaging or spectroscopy in the torso of a subject as large as a human, then of course, I thought, let's go back to heart because we have higher signal to noise and we can do much better. And we have not been pushing phosphorus spectroscopy so far in the heart at 7 Tesla, but people from Oxford, you know, who worked with us for a while, we helped them a lot, we got them going, and now they are doing very good phosphorus spectroscopy in the human heart at Oxford University.
So I'd like to ask you kind of a very forward-looking question. Can you imagine science or technology beyond bold imaging, that is a physics that doesn't have the same limitations in terms of temporal resolution and so forth that we currently face?
Yes, I can imagine that. And you know, one thing you learn in MR is never to say never, because MR, magnetic resonance, has been so dynamic. Every year there are new measurement capabilities that come in, and so I can imagine that. And in principle, you know, people have been cognizant of the fact that neuronal currents will cause signal dephasing, and you can detect it. And then every once in a while, some reports come in saying, "Oh yeah, I finally detected the neuronal currents, but you know, they are weak effects or they're not reproducible." And but there are some theoretical reasons for being sensitive to all of these phenomena that goes on in the brain. So I think one day somebody's going to figure out how to tap into that, you know, without in a robust way, let's say, and then come up with a new way of imaging brain function besides both. And of course, people are already, there are claims, for example, that diffusion imaging is sensitive to brain activity. This is still not currents, but maybe potentially closer. But I think that, you know, I really, I mean, when somebody comes up and says, I finally imaged neuronal currents with MR, I look at it with careful skepticism, but yet I'm optimistic about the future of this development.
Thank you.
Does that high field have any effect on the physiology of the person who gets this scan? Like, have an affect them physically in any way?
That's a that's a question that we always ask. We worry about. So far, nothing we can tell. You know, of course at this point, thousands of people have gone through 7 Tesla. And then because we did the very first 7 Tesla, we actually did what we did. We are doing for 10.5 Tesla now. In other words, it was not, we couldn't, I won't go into the details, but we had to work with FDA and get an IND, IDN, or investigation of the IDE, investigation of the device exemption. So we had to get an IDE. So you work with FDA, and so what we do first to get that, we got that for the 4 Tesla and the 7 Tesla and now 10.5 Tesla. And to get that, what you do is you do a lot of animal experiments first. Luckily, animal magnets, because they are smaller, they can go to higher fields. So when we actually had the 7 Tesla, we had a 9.4 Tesla animal system. And we did a lot of studies with rats who they spent zillions of hours in the magnet. They actually, the females were pregnant, and they were in the magnet when they were pregnant. So the pups actually, a lot of the development occurred in the magnet. And we did histology. We did behavior. Can they go through the maze as good as the controls, and et cetera, et cetera. So we published that paper. The only positive conclusion of that paper was female rats are smarter than male rats, but that was about that was it. There was no effect of the magnetic field, and so we have done this now at 10.5 Tesla, and then you get the IDE. You do a lot of testing now. We are doing it at 10.5 Tesla. These people are really thoroughly checked for behavior, vestibular function.
Vestibular function.
Yeah.
Do people get dizzy when they go in?
People always get dizzy to a certain degree around magnets. This is a real phenomena. It's not. Some people are more sensitive to it than others, and whether they get dizzy if they actually are going slowly into the magnet is a different question. And I don't have the. I don't have a. We are in the process of doing this, so I don't have a rigorous answer for you yet, population average, so to speak, soon. But what I can tell you is that we actually, when we had the 4 Tesla, we did a very interesting experiment. Because you give people a questionnaire, or not a questionnaire, they have to actually read IRB document, right? And then it says you can feel dizzy, you can feel this, you can feel that. You can feel phosphines and see phosphines, etc. And then we were curious how much of that was caused. When people come out and we give them a survey, we're curious how much of it is caused by the suggestion. Is it real or not? And one time the magnet was down for repairs. It was at 0 Tesla, and we actually ran through the experiment exactly like, okay, come on in, you have to be careful, all right, do you have any metal? Oh, I got a, metal thing on my belly button. Oh, we got to take that out. And take that out. Put the subjects into the magnet. We had recorded the sounds of the gradient. Okay, now the session is starting, don't move, everything like that. And then we give them at the end a report, a card saying, okay, please can you fill out this? And we looked at 0 Tesla, 4 Tesla, and about 25 to 30% of the subjects felt the world was upside down. They felt this, they felt that. Whether it was 0 Tesla or you couldn't tell them apart. So, anecdotes are there, and there are some effects that are real, but not all effects are real.
I want to take the opportunity to ask you not really scientific question, but you are in the unique position of pushing the envelope and leading the field for 40 years, 4 decades.
I'm that old.
From 1980s until now. So usually a leader, a pioneer only lasts for 10 years. When field mature, the person retire. But you have been leading the field, hardened brain and engineer for 40 years. Do you have any advice for the young people, lineages here? Any advice to the students?
Eat a lot of blueberries. Ride your bicycle to work. Never watch television, and always stay curious. So.